Well, the species of the month is the Sara Orangetip, and we haven't, of course it's a very common butterfly. We see it all the time on our field trips, but believe it or not, the last time we talked about Sara Orangetip here was 10 years ago, and that wasn't me either. That was another substitute speaker. I thought we were Sara, the Sara Orangetip, one of our little jewels that comes out very early in the spring. It's single-brooded, pardon my voice, it's single-brooded and overwinters as a pupa, and so it's matured within the pupal shell, and so first thing in the spring, as soon as you get decent weather, you see these little pierrots popping out. The eggs are typical pierrot eggs. They're kind of pointed on both ends. Interesting thing about pierrot eggs, you notice the size of this one, 1.5 millimeter. And when we look at the first instar larva, what's the size of that? 1.6. It's about the same size. Just about any other butterfly, when it comes out of the egg, because they're coiled up in the rounder shaped eggs, when they hatch, they're roughly twice as long as the egg. But the pierrots in this cylindrical egg, they're not coiled up. They're kind of straight in there. So when they come out, they're about the same size. Interesting thing about pierrots, most of them actually, not just this one. You notice the little tips of liquid on the tops of the spines. Those that's a chemical. It's actually a deterrent chemical. And David James has done some work on analyzing those little droplets, and it appears that one of the chemicals that's in these drops is capable of repelling ants. So
do we know what it is yet?
Yeah, he gave it a name, but it's
Hydropyridoxylene something or other.
methyl bad stuff.
Yeah.
I don't remember. I can get you the name that he came up with. In fact, I think he put it in our book. This is the second instar, and a couple things to notice about that. Well, first of all, you can still see a few droplets around this. They're pretty much gone at this stage. But notice the shoulder here, the front end of the larva. Anybody know why it's so swollen there? What's going on?
But ready to bust.
It's getting ready to bust. If you look at the head capsule, now the head capsule, you might remember, is rigid. The body stretches like crazy. It can stretch so that the larva can grow to about twice its size before the skin reaches its elastic limit. But the head is rigid. It doesn't change throughout the growth of the instar. And you see, it looks kind of white on the back, kind of odd. And that's because the larva inside has already pulled its head out of that head capsule, and it's stuffed in this big bulge here. And it's just getting ready to bust out. Now that's 6 millimeters, and here's what it looks like after it busts out. About the same size, 6.5 millimeters. Notice proportionally how big the head is now. And if we look at the previous slide again, proportionally the head is much smaller because it's an old second instar head capsule, and this is the third instar head capsule, proportionately large. So that's one way you can tell when a larva has molted. By the way, this is the old molded skin right here.
Do you know what host that is there?
I'm sorry?
Do you know what host we're seeing? Host plant?
Well, it was an Erebus. I don't remember which one I was wearing.
Look at the spiky hairs, man.
It's got the stellate hairs, the
Yeah.
hairs that radiate from the multiple hairs coming from one base. As a star.
Maybe. They look pretty simple. Still look like they're pretty single at this stage. But maybe so.
Look at the previous slide. We're talking about the Erebus, not the caterpillar.
Oh, okay.
Yeah, see those spiky, that's different.
Okay. Like they're, oh man. Push the wrong button here. You mean like
Yeah,
this? Yeah, okay.
I think they're mini ninja stars myself.
Okay,
The little droplets came back.
yeah, I was noticing that the little droplets aren't present again at the tip, so maybe they kind of dried up in the old instar shell before it split out and came up to this new third instar. Here's later in the third Sara. It looks quite a bit different. It's changed to a much more. Okay, it's changed to a much more camouflage color. Notice how well it camouflages against the green Arabis. And when you're seeing it, especially from the back is this darker green, and they line up with the leaves, and they can be quite hard to see. And here's the 4th inch star.
Is that color, the pale color at the lower end of that on the laterally, is it kind of a pinkish color? Or?
Back here?
Yeah.
Maybe a little bit, but I've never really called it pink. One thing that starts developing at this point, notice how the white stripe on the side goes clear up onto the head capsule. You see that?
Yeah.
And we'll see that more distinctly on the next slide here. You see how it goes right onto the head capsule. We have a desert marble at Lada, which has larvae that are very, very similar to the Sara Orangetip, except you can easily tell them apart because on Lada, that white is not there. The white goes right up to the head and stops. So they're pretty easily separated, even though they're very similar larvae. The pupae are very distinctive. They have this big bulge in mid-body and then the points on each end. Cremaster here holding it at the bottom, and then of course the girdle that holds it. This is the form that it's going to be in throughout the winter. And initially the pupa is green like this, but as it matures, it turns brown. And remember, it's going to be overwintering, and so it's going to be in brown sticks. And so as time goes on, it blends better and better during the winter, so it can avoid detection by predators. Here again is our...
Hey.
Yes?
So if they emerge in the spring and they only have one brood,
They just have one brood.
so when do they develop the pupa?
The pupa, it depends on what elevation, but the butterflies fly early in the year, in May probably, and the pupae will probably be forming around the end of May. So
they
all
last a
through
long
summer.
time.
Oh, yeah. Well, they can last actually a couple of years.
Yeah, a lot of our butterflies can, if you stress them, they can wait over an extra year.
And I was just looking at that thin little bit of silk thread,
Uh-huh.
and they're lasting all that time, and that holds them in the wind.
Sometimes not.
Yeah, sometimes they break, and
Just
sometimes
kind
they
of
fall.
imagine.
We found one in a leaf litter, Rod Crawford was straining leaf litter, and he came up to me with this thing, he says, "This looks like an insectoid thing," and it was actually a marble, euchloe.
Oh, okay. Well, you, Chloe, do
But it's
basically
all-
the same thing.
Yeah, and it emerged, so it was fine.
Here's David James's prize-winning side, underside of the Sara Orangetip. He actually won a photo contest with this. Beautiful.
How many people think there's actually green in that image?
It looks green to me.
There's no green at all. It's all yellow and black, I guarantee you. It's an optical illusion. Butterfly wings are deceptive.
Oh, you mean the yellow on the black?
Yeah, yellow and black. There's no green at all. No physical pigment of green.
That's a freshly emerged individual. Of course, he reared it, but it's just in pristine condition.
Is that why it's so hairy, fuzzy on the head?
Well, yeah, partly. The hairs kind of wear off as they get older, but yeah, that's just prime fresh out of the pupa. One of their favorite postplants, they do like a whole range of crucifers, but one of their favorites is this purple flowering crucifer. Anybody know what this is called?
I thought this was Pinatorum.
I mean, the genus.
Oh, Arabs.
Arabis.
Well, it was Arabis.
was.
Yeah, shut up. You're just making trouble.
Oh, I have it right up there. I'm sorry.
Yeah.
Anyway,
That was a good guess.
Anyway, Arabis is not an abundant plant. You see it all over the shrub steppe, but it's kind of everywhere in small numbers. And the Pierrins don't seem to be real fussy about which Aramis they use. Here's another Aramis. This one I've tentatively identified. By the way, notice the genus name Bucher.
Well, that looks like butcher.
Well, however you
Butcher
pronounce
of the Arabs.
And the plant folks have done a real job on Aramis. They've just butchered the terminology, so everything's been split and renamed, and new species erected.
I just want to clarify. I said, oh, this site in Seattle has this, the last patch of Arabis or Pteritis glabra, and I neglected to mention that it's a preferred host plant of the Sara Orangetip. But I implied that it's a rare thing in this area.
Yeah.
They're really good to eat too. I mean, I like them just as much as the...
Them being a certain species or any old Arabis?
Any old Arabs.
Okay.
And just about anywhere you find a reasonably fresh Aramis, this one is not terribly fresh. You can see it's kind of wheeling down, but it's still enough to attract butterflies. Whenever you find a little patch of Aramis, if you just sit there and watch for a while, sooner or later, the Sara Orangetip's going to show up on it. And this, of course, is a male. By the way, I should have mentioned when we looked at the adult pictures that the female, where the male is white, the female is bright yellow. Otherwise, they're pretty similar. Names. There's a lot of confusion about names in the literature. First of all, there are only two species of Anthocris in America. The desert orangetip and the Sara Orangetip are the only two. And the desert orangetip is pretty easy to separate. There's a number of differences, but the main thing you're going to see is that the orange patch on the tip of the wing, it's only about half as big in the desert orangetip as it is in Sara. It starts at the front of the wing and only goes halfway back, whereas Sara goes all the way to the hind part of the wing. According to Mr. Pelham's catalog, there are 14 subspecies of Sara in America. With this many subspecies, one thing you can kind of instantly recognize is there's a lot of variation in the Sara Orangetip. Because of this, there's been a lot of folks that have been looking at whether it should be split. And in fact, many of us have the Guppy and Shepherd butterflies of British Columbia, very nice book. But they did split the Sara. They split it right down the Cascades. Everything on the west side they continue to call Sara, and everything on the east side they call Stella Orangetip. But as indicated there on the last line, the split has been pretty well discredited because the criteria that they used to split them just didn't stand up. But there's another split that's in the works, and maybe John, maybe you can talk about this. This is what Todd Stout's working on.
Yeah, our mutual friend Todd Stout out of Utah has been working on this group for over a decade. He contributed a great deal to my understanding, and it was cited several times in the catalog. It seems obvious there's at least two distinct species in this complex, and very probably three. It's very difficult because in the areas where it's critical, like in the southwest desert, basin and range areas, and so there's large areas where there are no orange tips at all. So trying to find relationships, phyletically between these kinds of and these kinds of distributions is tough. But he's found very, very profound differences in the life history, and of course that's where Dave and Todd really come together on the Daves and Todd. They've really been looking at Anthocris. You've gotten a lot of images that Todd has examined closely. So it's hard when you have something that's basically so similar over a large range. It's difficult when people start looking at dividing it into species, and then you see one attempt already failed, and that was basically because of a miscomprehension of the name Stella. Stella is just a name for a Californian population, and it got off to a bad start, and it proliferated, and it's like that happens in popular works a lot of times. It was an opera that really spread that name around. But I'd say probably a year now. Probably within a year I'll have his paper out. I've already looked at it, and it's being evaluated by reviews. So. Is
this name correct? Julia is what will happen?
No, that's right. Yeah, ours would be Julia. Julia is the name of the Coloradoan orange tip, and the reason that we ended up with that is that Julia is a very old name. I mean, it's the next oldest name to Sara itself, and the name Sara's orange tip is basically a Californian insect. hardly gets outside of the state of California, barely into southwest Oregon, and it almost always is two brooded over most of its range anyway. It's two brooded, and actually biologically is a rather distinct entity. That it's taken us so long to find this out is because, you know, we've become accustomed. Everyone tells us, you know, that Anthocris Sara, Sara's Orangetip, you know, Anthocris Sara, Sara's Orangetip, and you know, you don't look further sometimes.
So anyway, by the field season this next year, maybe we'll no longer have Sara's Orangetip, we'll have Julia's Orangetip.
You did say our Orangetip we have. You're talking, Julia would be the same species east and west of the Cascades.
Right.
Thank you.
Yes.
And John,
Uh,
what about DNA testing? How do you use that to distinguish?
DNA has a lot, it's a really difficult question to answer. With taxa that are actually closely related, and in fact, you know, lineage sorting is impossible where the hybridization takes place. And some taxa hybridize at a regular rate. It's just a functional part of their biology. So what happens though is that in mitochondrial genes, you know, this you know, lineage sorting and switching really creates problems. You can't get really good data off of the two genes that are used most often, the carboxylase and alpha elongation. I think they're both mitochondrial genes. And that's where I mean, you look for differences where things are variable. You don't compare things that are the same. So the problem is when you have hybridization complicating it, it doesn't, you know, DNA hasn't been really useful at all here. So good luck. I mean, you know, we did have DNA 20 years ago. We still don't, so what's the loss?
Any other questions?
When you see a female orange tip on the wing, how do you tell it from marble?
Well, I don't think you'd mistake them too much seeing them fly. The marble looks white in flight, and the female Sarah's going to look bright yellow, flashing bright yellow. I don't think you're too hard to tell apart, you do.
Well, there's two marbles. One is quite different from the other one. So
Yeah.
I think Asanides probably you could convince, you could make a mistake
Yeah.
between, but they don't have any orange tip. I mean, that's the number one thing.
Yeah, the females have an orange tip as well.
Yeah.
So you can see that orange flashing, but
Now, the eastern orange tip, the females don't have orange tip, so that would be appropriate. You know, they look like a marble.
and yeah, the large marble does have a lot of gold on the underside of its wing, so it could give you the impression of yellow. Okay.
Okay, thank you.